Bob Weinberg. My lab is at the Whitehead Institute of MIT, and my research has been focused on oncogenes, tumor suppressor genes, and over the last 2 decades, more on the stages of malignant progression. That is to say, how primary tumor cells actually succeed in becoming invasive, malignant, and ultimately spawning metastases. Before the millennium, our focus, like that of many other labs, was focused very centrally on the genetic determinants of cancer pathogenesis. That is to say, oncogenes, tumor suppressor genes, DNA repair genes. But at the beginning of the new millennium, it became apparent to us that genetic paradigm did not really apply in any serious way to the last stages of cancer development, that is to say, the malignant progression that empowers primary cancer cells to become invasive, ultimately to disseminate via the circulation, and to seed new cell colonies, cancer cell colonies in distant sites, that is to say, metastases. Indeed, to my knowledge, no one has ever actually found mutant genes that are closely correlated with the metastatic behavior of cancer cells. And so we began to work on that area and became interested in the cell biological program termed the epithelial mesenchymal progression, or EMT, epithelial mesenchymal transition, which exists in a latent form in epithelial tissues and is activated upon a wound healing. And the activation of that program, the exploitation opportunistically of the EMT process, is what empowers primary carcinoma cells to execute most of the steps of the invasion metastasis cascade. Most recently, we've been interested in some of the last steps of this cascade. After carcinoma cells disseminate to distant tissue, they often go into a dormant state where they persist for extended periods of time, remaining viable, and only with the passage of time may some of them become activated, that is to say, awakened, and begin to seed clinical relapses, which one sees in most dramatic form, for example, in breast cancer patients who've previously been deemed cured of their tumor, only to experience or suffer from metastatic relapse 5-10 years after the initial tumor was successfully removed. In terms of my original association with the cancer research field, I received my undergraduate and graduate training at MIT. After doing 2 postdocs, I came back to MIT, having been part of the original MIT Cancer Center, and I was closely affiliated at the time with David Baltimore. He was interested in how retroviruses replicate themselves, that is to say, the enzymology of reverse transcription, and I became interested, in fact, in how the DNA molecules that are synthesized in recently infected cells are produced and how they function in terms of enabling the viral growth cycle. Of note, by the way, is the fact that the replication of retroviruses was only incidentally involved in the whole process of cancer formation. However, we began to study in my own lab Harvey sarcoma virus, which is a transforming retrovirus, and through that route became interested in the mechanisms by which normal cells, after being exposed to a mutagenic carcinogen, then become neoplastic. And this led us, in turn, to attempting to understand the origins of the transformed state of normal cells that have been transformed to a neoplastic state following exposure to, as mentioned, mutagenic carcinogens. This led us, for example, in 1969 to describe the existence of mutant transforming genes within the genomes of cells that had been transformed through exposure to methylcalanthine, and in turn to the discovery that these genes were actually the same as genes that had previously been discovered in the context of retrovirology. And through that roundabout route, we concluded that the repertoire of genes that were discovered through the techniques and genetic analyses of transforming retroviruses, the same bank of normal cellular genes that are kidnapped by these retroviral genomes, the same genes also serve as target for malignant transformation at the hands of mutagenic carcinogens. And so that line of work has characterized much of what I've done in recent years. Uh, it happens through sheer happenstance that I was born at a time when, as a student, I could enter into the then newly minted field of molecular biology. I remember as an undergraduate hearing about the triplet code and how DNA makes RNA, makes proteins. And that led me to conclude, as obviously many other people did, that one could understand the totality of the biosphere by following this paradigm. DNA makes RNA, makes proteins. Through that, I concluded one could understand virtually everything there was to know about life on this planet. And that continues to be basically the case. But obviously, it was a very exciting time, through my good luck, to be present when all this was happening. And especially at MIT, which had early on taken on the theme, the crusade of molecular biology. I had the opportunity to interact with people and learn from people who were involved in the then nascent field of molecular biology, gene cloning, and subsequently the molecular bases of cancer formation. So, through much luck, I was very, very fortunate to be able to associate with people who were unraveling for the first time the genetic basis of cancer. And it's been a very exciting run being associated in the Cambridge, Massachusetts biomedical research community with so many exciting, young, and interesting researchers.